Thank you. Okay, well, thank you very much. So when I was a young man, I thought I was going to be a clinical doctor. Here I am on the first day of internship at Johns Hopkins. My chairman was the great Victor McCusick, who not only was a member of the society, but also had won the Benjamin Franklin Medal. And he instilled in me an appreciation of the power of human genetics. Francis, I'm sure that would resonate with you, as well as the power of understanding the work that's been done before you, having some sense of medical history. I went, as you heard, to the Dana-Farber Cancer Institute, again, expecting to be a clinical oncologist, but I had the very good fortune of meeting the great David Livingstone, who sadly we lost 2 years ago. And it was David who inspired me to be a scientist and taught me how to be a scientist, so I really owe my scientific career to David Livingstone. Now, I understand this is a very sophisticated audience, but I suspect it's also a fairly diverse audience, and I always tell students no one's ever gotten mad at me because I made them feel smart. So we're going to have a 3-slide primer on molecular biology to make sure everyone's in the same boat. So if we recognize that a segment of DNA has the instructions for making a protein, we refer to that as a gene. You can think of that as the master blueprint. If the cell needs to make more of this protein, it makes copies of the DNA in the form of RNA. So you can think of these as the Xerox copies of the master blueprint, and then these get sent to the factory floor to make the protein using those instructions. And a protein I like to think of as sort of a string of pearls, where each pearl is one of 20 potential building blocks, so-called amino acids. And it's the 3-dimensional folding of that string that determines the function of the protein. And we represent these different building blocks with different letters, such as the one shown here. Now, the problem is sometimes the DNA sequence gets altered or mutated. This misinformation gets incorporated into the RNA, which in turn, at times, can lead to a disastrous change in the protein. So here, the building block represented by the C on the left has been changed to the building block represented by the F. Now that might seem like a subtle change, but this is literally a mutation which in certain families around the world predisposes them to various cancers. And then the last part of the primer is that genes can be, if you want, on or off, or scientists say expressed or not expressed. And so if the cell needs more copies of that protein, it will turn on the Xerox machine and send those blueprint copies down to the factory floor. And that's under the control of so-called transcription factors. Now, in David Livingstone's lab, I had worked on a so-called tumor suppressor gene, the retinoblastoma gene. This is a gene that contributes to cancer through its inactivation. And then, as you heard, I started my own laboratory in 1992. And shortly after starting my laboratory, this paper crossed my desk, which was the cloning or isolation of another so-called tumor suppressor gene, in this case, the von Hippel-Lindau tumor suppressor gene. And this is a gene that, when altered and passed down from generation to generation, can predispose individuals to various cancers. And I should point out that this work was led by investigators at the NIH, which sadly is currently under attack. Now, what do these families look like? So here is a number of these von Hippel-Lindau disease families that have been followed for years at the National Cancer Institute. Every filled circle or square is an individual in that family that's developed one of several different types of cancer. And the most common cancers include kidney cancers, blood vessel tumors called hemangioblastomas of the eye, brain, and spinal cord, as well as so-called neural crest tumors, sometimes referred to as paragangliomas. Now, this disease affects about 1 in 35,000 people around the world. It was described by the German ophthalmologist Eugen von Hippel in 1904, and subsequently reported in greater detail by the Swedish neuropathologist Arvid Lindau. Now, just to look a little more closely, your DNA is organized into 23 pairs of chromosomes, but here I'm showing you the pair of chromosomes from mom and dad that harbors the VHL gene. So people with von Hippel-Lindau disease have inherited a defective version of the VHL gene from mom or dad. In this schematic, it's the maternal copy that's defective. But unfortunately, being born like this, they have a 90% chance that one or more susceptible cells in their body, such as in the kidney or in the eye, will spontaneously mutate or lose entirely the other normal copy. And that's the cell that's then at risk of going on to form a tumor. And as you would expect from the knowledge that inheriting a defective version of the VHL gene predisposes to, for example, kidney cancer, if you now look at garden variety non-hereditary kidney cancer, and the most common form of kidney cancer is so-called clear cell renal cell carcinoma, you again see that frequently both the maternal and the paternal copies of the VHL gene have been mutated or lost. But here, both mutational events, or hits, if you will, have occurred sometime after conception, in contrast to VHL disease where the first mutation has been inherited from mom or dad. Now, I knew from my clinical training that the tumors seen in von Hippel-Lindau disease are notoriously rich in new blood vessels, and that's because they sometimes overproduce various growth factors such as vascular endothelial growth factor, which acts as a fertilizer for blood vessels. And then another curiosity is they sometimes cause patients to make too many red blood cells, and that's because these tumors sometimes inappropriately produce a hormone called erythropoietin, which stimulates red cell production. Now, what VEGF and erythropoietin have in common is that they're normally induced when cells or tissues are not getting enough oxygen. And this makes sense. If you're not getting enough oxygen, you want to be able to improve oxygen delivery, you want to improve the oxygen carrying capacity of the blood by making more red blood cells, and you want to have more blood vessels supplying those red blood cells to the tissue in need. So this was the clue that by studying the VHL gene and its protein product, we might actually finally begin to understand how cells sense and respond to changes in oxygen, which seemed like an important problem since oxygen plays such an important role in so many diseases, including cancer, but also heart attack, stroke, etc. Now, we knew from the work of many laboratories, including the work of my fellow laureates, Greg Semenza and Peter Ratcliffe, that many of these so-called hypoxia-inducible genes, and hypoxia is just a fancy word for low oxygen, such as VEGF and erythropoietin that I just mentioned, are under the control of a transcription factor, but you're now experts on transcription factors, that contains two subunits, the alpha subunit and the beta subunit. And so this is going to turn on the Xerox machine. But we also knew from their work that the alpha subunit is normally destroyed or degraded if oxygen is plentiful. It only becomes stable and active when oxygen levels are low, hence hypoxia-inducible factor. And we and others then went on to show, and I think this is hopefully the most complicated slide I'm going to show, that the VHL protein is actually part of a multi-protein complex that, in the presence of oxygen, chemically attaches a little flag made up of a mini protein called ubiquitin. And by adding this little flag, the HIF-alpha gets targeted for destruction by a cellular garbage disposal we refer to as the proteasome. Whereas when oxygen levels are low, or the VHL protein has been mutated, such as in kidney cancer, now HIF-alpha can accumulate, dimerize, or bind to its partner protein HIF-beta and activate genes such as VEGF. Now, this was very satisfying, but it still begged the question, how do cells know, if you will, whether oxygen is or is not available? How would the VHL protein know whether it should leave HIF-alpha alone or whether it should target it for destruction? And our lab and Sir Peter Ratcliffe's lab working in parallel showed that in the presence of oxygen, one of those building blocks within the HIF-alpha protein that we recognize by P for proline gets chemically modified. A little hydroxyl group gets added to the proline, and that serves as the signal that oxygen actually is available, and this then generates a binding site for the VHL protein. And what was very satisfying about this is even a sixth grader would appreciate that this O stands for oxygen. So this was an immediate link to oxygen availability. Now, of course, like most things in biology, it's a little bit more nuanced than that. We and others showed that the proteins that add that hydroxyl group, and by the way, if a protein accelerates a chemical reaction, we refer to it as an enzyme. So the enzymes that do this work, which have this name of Kaelin, they split molecular oxygen, and they use one of the oxygen atoms to add the hydroxyl group. But importantly, they also have a fairly low oxygen affinity, so they're very sensitive to changes in oxygen availability in a physiologically relevant range. Now, these enzymes also require several other chemicals. They require iron, but I also want to point out they require this funny chemical over here called 2-oxyglutarate, which during the reaction gets modified and converted to succinate. Now, I point this out because when we first discovered this 20 years ago, it immediately became apparent that you could inhibit these enzymes with orally available drug-like molecules that would compete with 2-oxyglutarate and effectively shut these enzymes down. And once you shut the enzymes down, the body or the tissue thinks it's not getting enough oxygen and responds accordingly. So, for example, if you take this by pill form, your body thinks it's at the top of Mount Everest, and you start making your own red blood cells because you start turning on the genes dedicated to red blood cell production. So a number of pharmaceutical companies picked up on this and started to develop these orally available drugs that would basically act as hypoxia mimetics. You would now think you're not getting enough oxygen. And the initial indication was an anemia, where you're not making enough red blood cells. And there are millions of people around the planet who have anemia because of chronic kidney disease. And for that and other reasons, they don't make enough of their own red blood cells. Now, the first of these to be approved was a drug called daprodustat. I've been involved with a company called Fibergen that was developing a drug called roxadustat, and I should declare I have a financial conflict of interest in roxadustat. Suffice it to say that this drug has been approved in over 44 countries around the world. Sadly for me, the US FDA decided they wanted yet one more phase 3 safety study, and the company figured out that by the time this trial ended, the drug was going to be off patent. So that's a whole other discussion. Importantly, for a variety of reasons, the first country to approve this drug was China, in part because of concerns about the reliability of their transfusion supply, et cetera, et cetera. And so as of last month, there are several 100,000 Chinese patients who take this drug, roxadustat, every day to treat their anemia as an alternative to transfusions and erythropoietin. Now, returning to the role of HIF in cancer, what I didn't tell you was HIF's actually a small family, and I show you the two most famous members of the family here, HIF1 alpha and HIF2 alpha. But work that we and others did showed that at least in kidney cancer, the culprit really is HIF2. And in fact, many kidney cancers get rid of HIF1 altogether. So what could we do about this therapeutically? Well, by the 90s, a number of drug companies were already making inhibitors of VEGF. And we argued that if VEGF inhibitors were going to work in any solid tumor, they should work in kidney cancer because of this direct and intimate connection between HIF and VEGF and VHL. And that turned out to be the case. So I think we're up to 8 or 9 VEGF inhibitors that are now approved for the treatment of kidney cancer. So that's the good news. But the bad news is not every patient with kidney cancer will respond to these drugs. And even those that do usually will progress within a year or two. So how can you do better? Well, I think you might argue based on first principles, rather than targeting any one single HIF2 target gene, maybe we should target HIF2 itself. But the conventional wisdom was that HIF2 wasn't druggable. It's an intracellular protein, and it was thought to lack the right kinds of nooks and crannies to drug with a small molecule. But fortunately, two wonderful scientists who were then at UT Southwestern, Rick Bruick and Kevin Gardner, ignored that dogma. They identified a potentially druggable pocket in the so-called PAS-B domain of HIF2 alpha, which is what I'm showing you here. They then, in their academic labs, identified a chemical that could bind to this pocket, and in so doing, cause the protein to refold slightly in such a way that it would no longer bind to DNA. Now, these were good for proof of principle, but they had many blemishes and warts that would have prevented them from being true drugs. But fortunately, they were out-licensed to a company called Peloton Therapeutics that was later acquired by Merck. And again, I declare another financial conflict of interest. But the medicinal chemists at Peloton made these chemicals much more drug-like. They improved their potency, their specificity, and their bioavailability. Our lab and the lab of my former postdoc, Jim Bruguerolas, then showed that these first-generation HIF2 inhibitors were highly active in preclinical models of human kidney cancer. An improved version of this molecule called belzutifan then went into phase 3 testing and ultimately was approved for the treatment of advanced renal cell carcinoma. Now, a truism, and by the way, I cannot see a clock. I'm told there's a clock there, but this is dangerous. But I also speak New York fast, so I'm probably doing okay. So the drug was approved for sporadic, non-hereditary kidney cancer. But a truism in medical oncology is that most cancer drugs work better in a frontline setting than they do in very late lines of therapy. And so we were able to convince Peloton and then Merck to treat 61 patients with von Hippel-Lindau disease who had measurable kidney tumors that had never been treated before. These patients develop so many tumors over time that they're often put in surveillance programs where they'll have an MRI every 3 or 4 months. And only if a tumor changes in a worrisome way will they go to the operating room, whether that's a kidney tumor or an eye tumor or a spinal cord tumor. So these are so-called swimmers plots for obvious reasons. So here are the 61 patients. So each horizontal bar is a patient on the trial, and how long they were on the trial with the HIF-2 inhibitor at the time of this analysis. So for orientation, here's 1 year on therapy. All the patients with the black arrows were still doing well on therapy at 3 or 4 years. And every place you see a blue square or a green square, that patient's kidney tumor has actually shrunk or disappeared altogether. Now, if you prefer so-called waterfall plots, here's how the tumor shrinkage looks on a waterfall plot. So again, here are the 61 indicator tumors that allowed these patients to get on the trial. Every time you see the bar go below 0, the tumor is shrinking on the drug, and every time it goes above 0, it's getting larger. I'm a lumper, not a shrinker, not a splitter. So I would say all the tumors got somewhat smaller, but if you're a splitter and you like RECIST criteria, the formal RECIST response rate was 60%. Now, to be on the trial, you had to have a measurable kidney tumor, but you could have any of the other tumors that VHL patients develop. So, for example, some of these patients had central nervous system hemangioblastomas, and you can see they responded very nicely. Some of those patients had those neuroendocrine tumors I talked about, and you can see those tumors responded as well. And perhaps most importantly, I mentioned these patients were in surveillance programs. So in gray are the 4 years of observation for these patients before they went on the HIF-2 inhibitor in green. And every place you see a little geometrical symbol, that patient's going back to the operating room to have another tumor removed from their kidney, their eye, their spinal cord, to the brain. And you can see that the surgeries, they don't go to 0, but they certainly decrease dramatically. And so based just on this phase 2 trial, in the 61 patients, the FDA did approve this treatment for von Hippel-Lindau disease almost 100 years after the initial clinical description. Now, it's very easy to look at clinical data and forget that there are actual human beings attached to all the data, at least it is sometimes for me. So I thought I would just show you one social media post. It was sort of fun. My kids taught me how to do this. I didn't have to wait for the New England Journal article. All I had to do was go to social media sites because I could see the patients were posting that they were responding. Because you can imagine if you have von Hippel-Lindau disease, you've had the sword of Damocles over your head. And you assume this disease was going to be your undoing. But here's a typical social media post. I never thought I'd see this day, and they're describing that their tumors were getting smaller or were stable, or in some cases had disappeared entirely. Now, in closing, I showed you this paper earlier, Eugen von Hippel's paper in 1904, but I also told you Victor McKusick taught me the power of understanding medical history, and it was also Victor McKusick who pointed out this was not, in hindsight, the first description of von Hippel-Lindau disease. The first description of von Hippel-Lindau disease actually occurred 10 years earlier from the great English geneticist Treacher Collins. So why was this paper largely overlooked? Why was it largely ignored? There was a simple reason. It was published in English. So this was the great descriptive era in medicine. And if you could only read one medical literature in the late 19th century, or the early 20th century, you read the German literature. So ironically, his undoing was he published in English. Now, what changed? Well, one thing that changed, of course, was World War II and the demise of Germany. But the other, I think, and I see this name comes up again later in the week, was the work of Vannevar Bush, who was also a member of this society. So Vannevar Bush, if you don't know, was arguably the key figure who, in the middle of the last century, convinced the federal government that the best thing they could do was to sort of pump prime by investing in basic and fundamental research in the academic sector, and then let the private sector decide when that knowledge had occasionally matured to the point where it was actually useful and could actually be applied. So this was absolutely brilliant. It served us well for decades. Now, I'd be the first to admit that Nobel Prizes don't necessarily perfectly capture a country's discovery power, but as a crude metric, you can see that during the descriptive era, when von Hippel and Lindau were publishing their papers, you could see the dominance of Germany, and then Vannevar Bush comes along, and something changes. And I think it's a combination of federal investment in basic and fundamental research, coupled, of course, with immigration, because we became this shining city on the hill. And especially with the brain drain and leaving war-torn parts of the country, people came here. Now, of course, we're all painfully aware that for some odd reason that escapes me, we're attempting to undo this. So this was my feeble attempt to add my voice to the many voices saying, what are we really doing here? Because again, we had a model which was the envy of the world, has served us well for decades, is responsible for our leadership in multiple innovation industries, including the biotech and pharma industry that I'm very close to, but certainly many other industries as well could map back to that investment in basic and fundamental research. So hopefully we can right the ship here, and I purposely don't want to leave you with a complete downer. So I'm always looking for something uplifting, something hopeful, so I'm going to just share one of many things that I think are hopeful. So this is a wonderful organization called the Environmental Voter Project. What they hypothesized and experimentally confirmed is that there are millions of Americans who never vote, who had they voted, would have voted with the environment as their number one issue. And these people, by and large, also believe in science and objective truth. Now, it also turns out how you vote is confidential. Whether you vote is not. So whether you vote is discoverable. So they can go into voter logs and look and see who votes, who doesn't vote. They've built incredibly accurate, robust analytical models to find these people. And by the way, if you think modern campaigning is about changing people's minds, that went out the days of the dodo. Modern campaigning is using analytics to find people who are already predisposed to vote the way you want them to vote. And then you use behavioral science methods to energize them and get them to vote. So this group actually does randomized clinical trials where they identify these non-voters. They then go in with these embarrassingly simple but effective behavioral science techniques, and they can actually map how many voters they have created in the last 10 years. So there are up to 2 million voters who never voted before they found them. They think they can get north of 5 million super voters, pro-environmentalists, pro-science, and at that point, you're more powerful than the NRA. Because the NRA is powerful simply because it's a galvanized block of voters that people can't ignore. So this is one path amongst many to try to get more people engaged in science, who believe in science, to actually vote. Because in the end, the solutions will have to be political. And my young people, friends and family tell me you have to have a QR code these days, so we even have a QR code. Thank you very much.
Thank you, Bill, for your work and for your comments. Questions? Francis. And state your name and where you're from.
Where I'm from. Well, let's see. I'm Francis Collins. I used to be at NIH. Bill, that was a great talk. Curious to know, with von Hippel-Lindau, obviously it's a two-hit model, but for most cancers, it takes more than 2 hits to get all the way to a cancer. Is there any other sort of additional mutation, a collaborator that might also be a drug target?
Yeah, I'm glad you asked that, because you've anticipated the answer. So yes, absolutely. So like most common solid tumors, I would say VHL inactivation is the initial key step. Some people even call it the gatekeeper. But you absolutely need additional mutations and other genes before you'll go all the way to a kidney cancer. We know that not only from studying the von Hippel-Lindau disease patients, but also looking now with modern techniques at the non-hereditary kidney cancers. And so I like to use the analogy of tumblers on a lock. And for kidney cancer, I think the first critical tumbler to fall in place is VHL loss, but there are at least 3 more. And we can talk offline, but we know most of those genes as well. This turned out to be really critical in considering the safety or lack thereof of drugs that stabilized HIF, right? Because naysayers would say, well, you're just going to have von Hippel-Lindau disease in a pill because you're deregulating HIF. Now you're smiling because you know that's an oversimplification, but you won't be surprised to hear that that was said by some well-intentioned people. But part of the issue is, again, you do need multiple other tumblers to fall into place before you'll be a kidney cancer.
Jack? Back there.
Jack Liu from East Lansing, Michigan. Thank you so much for this fantastic talk. I like everything you said, although I may not understand everything you said, but as an ecologist, I'm particularly like these slides. And because as an environment scientist, I think the interface between medical professional and scientists and the environment issues are hugely important. I'm wondering, how did you get, into these issues that I think are not just politically important, but also environmentally important? Thank you.
Yeah, so fortunately, I read the fellow who started this. His name's Nathaniel Stinnett, a very smart Yale grad who had worked in modern campaigning, knew how modern campaigns worked. And again, his epiphany was that if the environmental movement had the equivalent of the NRA, we'd be carbon neutral by now, because the politicians' behavior is so predictable and mappable, right? And he's the one who taught me things like, why the NRA does have so much power. And it's this galvanized block of voters. And he also taught me this simple fact that whether you vote is public. So he said, if you call your elected official complaining about something, the first thing they do is they have their assistant find out if you ever voted. If you've never voted, the conversation is over. It's over. Okay? If you do vote, they have very accurate data on what their constituents care about deeply. And for most of our lives, the environment has come in 8, 9, or 10, with 1, 2, 3 in some order being the economy, healthcare, and defense. So he realized he needed to move the environment up a bit. And again, I already told you the punchline. His hypothesis was absolutely confirmed that there are a lot of environmentalists who simply never vote. Now, the related question he gets asked, and I often get asked, is why aren't they voting? That's a whole other discussion, and I know we have some sociologists here. But there's a lot of overlap between these people. It's not, first of all, it's not a homogeneous group, but there's certainly a lot of overlap between these people and people who are historically suppressed from voting. So you may be solving two problems at once in terms of how do we get everyone engaged and voting.
Anyway, don't shoot me, but I'm a former U.S. Congressman, Patrick Murphy. But I'm on your team. So, the University of Pennsylvania. Listen, I follow this pretty closely, but of the last 10 elections, the 9 out of 10 were change elections, where either the White House, the House, or Senate changed. I love the fact that you're not saving this conversation for play company, that you're challenging all of us. Appreciate it. I would say to your thesis on this is that as long as you look like you're being part of the solution, saying, hey, listen, we need to change this, we need to do better, but look at the great success. I mean, I wrote it down, that we're killing the golden goose. So what do you think we need to do better as an organization at APS that cares about this, beside joining the environmental voter org, beside showing up today? Because it's easy to talk to the choir, but not the congregation.
Yes.
What would you say we should do publicly? I love the fact that you're on social media, but what other advice would you give us?
Yeah. Well, thank you. First of all, I actually like that you used the word team, because when I was a young boy, I didn't think this was a partisan issue. And when I give a longer version of this talk to young students thinking about a career in scientists, I often my first slide is usually Sputnik, since I was born in 1957. But in the 60s, 70s, and even largely in the 80s, it just was unfathomable that this ever would become a political issue. I thought we were all on the same team. And so how we get people to understand that some things should not be about partisanship, that we all get affected by these diseases, right? I wrote an editorial when my wife died of glioblastoma. She died about the same time as Senator McCain died of his glioblastoma. And I pointed out this is not a team thing. I mean, it's a team in that we're all on the same team. It's not Republican versus Democrat. But what happened during the 60s? Precisely because of Sputnik and the Cold War, there was bipartisan realization that this was almost existential, that we win when it came to science, technology, and engineering. So, when Obama said we need another Sputnik moment, maybe we need another Sputnik moment, but we need something like that. I mean, people have to understand that whether they're scientists or not, we're all in this together, and they all benefit from us being a center of innovation. And I can go on and on. So I'm preaching it to the choir, but I think it's partly education, outreach, as you said, crossing over when we can to our colleagues on the other side.
But we did have the COVID epidemic.
Yes.
And in fact, that separated us rather than bringing us together.
Yeah. Well, we were talking about this a little bit at lunch. I think sadly, and maybe it's in the unintended consequence book that we learned about this morning, I think people who are smarter about this than I am have pointed out that we're paying the price for people who, when it was expedient to do so, sowed distrust in the scientific method, whether it was people who made money from a carbon-based economy pushing back against climate change, whether it was the tobacco industry pushing back against the cancer risk, whether it was people who wanted to add lead to everything, including your gasoline and your paint. You know, the playbook was always the same. Question whether anything is objectifiably true, whether there really is such a thing as expertise. It's just a theory, it's your opinion. But we're paying that in spades, and I think the same playbook unfortunately played out a bit in COVID, although there was a bit of a stretch. Why? I don't completely understand, but I think that's where we got it. I'm sure part of it was the wishful thinking that we could ignore this and go back to work, because of course it was costly on multiple levels, right, to shut down.
Yep.
And so I'm sure there was some of the same economic interests, again, might have been behind some of it.
All right. Well, with that, let's thank Bill again.
Great. Okay.